I appreciate that. Yeah, we definitely. I'll talk about it more during the project updates, but we're definitely there's still a couple things figuring out, but I'm happy to at least say that we're in better shape than we were last week.
Well, that's as long as one's going forward, that's the most you could, I think, ask for.
That's fair. That's fair.
Well, I don't know. I mean, you could ask for more than that. But,
Momentum is the easiest, is usually the one thing that you can be sure you can deliver if something's going somewhere.
so, get the screen set up. I've been trying to go back and edit the videos. It's not so easy to, because when you do the exercises in class, having to go through and trim those out is a little bit, I mean, it doesn't have to be perfect. It's okay to have some dead air. But it's, it takes a little more time because you have to go in. But anyway, I don't know if anybody's ever looked at any of the recordings.
I look at them usually every week, in all honesty. I find it's one of those things that I find I'll get a decent chunk of it as we go through it, but sometimes, especially if it's been at the end of a long day, there will be one or two items that fly over my head, even if I don't realize it at the time. And it's been nice to be able to go back and kind of watch back through what we've done.
I mean, that is one advantage of doing things virtually, because if you do it in class, in person, the recordings are crappy. And I mean, it's not that I couldn't record the whole lecture separately, but that's an extra 3.5, 4 hours, which means it's not that I mind spending that time, but it's time I don't spend on something else for the class. And so I think at some level doing it virtually is not too bad. If you go back over any of them and you have suggestions for improving the abstract, or gee, you should edit out minutes such and such to such and such, please let me know, because it's easy enough to go back and clean those things up. Hayden has edit access to the account. Giuliano also does. So yeah, if there are things that, gee, I want my comments about the projects edited out or something like that, that's all fine. Just let me know what I can do to make them more useful. I know that they're certainly far from perfect, but I also know that these things, I like having some kind of resources. 3-hour single blocks are not the most useful kind. And I think YouTube does allow you to put indexes in, but I haven't figured out how to do that yet. So that's something that I probably have to learn.
Yeah, I mean, I was actually reflecting the other day because, I mean, you said at the start of the course, there's no actual like textbook purely dedicated to this. So in some ways, the video series is kind of a stand-in for the textbook.
Well, what I need to do more than anything is to take these slides and turn them into a Word document. And Microsoft does not make that easy. It's really a pain to go. Going from Word to PowerPoint is not so bad, but going the other direction is painful. And so really the way to do it would have been to write the lectures in Word and then turn them into slides rather than. Of course, there'll always be somebody who says, oh, you should just have done it in LaTeX. This is not my favorite.
LaTeX
Go ahead.
The outcome is beautiful. The time into it is like I had a class where the homeworks were taking, like 10-ish plus hours of like concrete work. And the one of the people was like, I would enjoy reading your homework more if it was all in LaTeX. And I did that a couple times, but the amount of time it took to translate all of the 10 hours into LaTeX meant sleep had to be sacrificed. And finally, I decided sleep was too important.
Well, I mean, I shouldn't say this too loudly, but people prefer to handwrite their homeworks and then take a screenshot, take a photograph of it. That's all right with me. I don't want people wasting time. I mean, I love that, but people do these nice type presentations. I'm very grateful to them and I enjoy them. But I don't want people spending more time than they need to. So if for example, if there's an analytic calculation, so you have some equations, taking a photograph of the equations and submitting that instead of typing it up in Microsoft Equation Editor or LaTeX or something else is okay with me. Certainly, as long as I can read it and I know what you're trying to do, that's all that matters. It's not the goal is definitely not to waste your time. There's always some way time wasting. There's no way around that in the world. In the world, there are always going to be some things that are a waste of time. But my goal is to try to help you spend as much time on the content of the course as possible and as little time on the surroundings. Now, for something like your class presentations, since that'll be recorded and presumably uploaded as part of the documentation for your application on NanoHub, it's worth spending time on the presentation. So those should look nice because they're going to be, I certainly could do better on my slide decks here too, but I try to make them look at least halfway decent. Since they're going to be up on YouTube, I want to make sure that anything that's persistent, you'd rather be as little, as you can't make it totally unembarrassing, but you want to have as few embarrassments as possible. I'm not sure where Giuliano is, but he's supposed to be giving part of the lecture today. So let's give him another minute or two. And then get started. As usual, we may not get through all of the material, but the deck's there. And I owe you a homework assignment, I know. I'm sure everybody is breathlessly waiting for the next homework assignment.
I think I also owe you a homework assignment. So.
Well, yeah, there's a little bit of that quid pro quo. There's as if people don't hand them in, and I tend not to give them up. But it doesn't always work like that, in the world. Sometimes the assignments keep coming. I know on my end, I keep getting assignments whether I finish the past one or not. Doesn't just because I was late doing it doesn't mean that I get to take a break.
Yeah, this week it was a matter of priority versus priority of the project versus the homework. And since we were meeting with Roland, I decided to shift pretty much everything into that bucket. But
right, I would always put the project first. I mean, ultimately, the projects are the meat of the course. The homeworks exist mainly to give you a practice and learning things that you need for the projects. Now, as far as the grading goes, if I remember right, and I'd have to look back at the rubric, the homeworks count a lot. And the reason the homeworks count a lot is because in my experience teaching this course, if people don't do the homeworks, then they find it very difficult to do the projects. But that's again, it's not, the expectation isn't that everybody gets a perfect score on every homework. It's more that people make a good effort. They try to learn from it. Okay. Next week, the plan is that I will give the lecture, but I have to have a medical procedure up in Indianapolis, and there's a small possibility that I won't be available to give the lecture next week. So we'll have Giuliano as a backup, and he'll focus on helping people figure out how to do uploads of projects to NanoHub if I'm not available. And he can also teach the antimony tellurium material that I've got here too, because he's TA'd that course a couple of times. Probably Joel could teach that material too. Joel and Jackson could teach that material. Okay, well, I think we'll get started then. Welcome back. It's lecture 12. My goodness, it means we only have 4 more weeks of the semester. I hope people's projects are coming along well. We've gotten through a lot of material. Today we'll continue, I hope, finishing up with chemotaxis. I want to talk about explicit forces. And if we have time, I'd like to start talking about loading subcellular models into cells to do biochemical regulation. And I know Joel and Jackson have been working on this, so they may have some ideas about this as well. And Giuliano is planning to give part of the lecture today, but if he's not here, it's all right. And as always, I have to remind you that we're being live streamed and recorded. And I was pleased to hear that from Connor that at least one person found the recordings useful. And I'll repeat that if you have suggestions, if you do look at the YouTubes and there are suggestions for edits in the videos or ways that I can make the documentation of the video better, please feel free to make those suggestions, and I'll do my best to go back and implement them, because there's always the possibility, there's always the opportunity to improve those presentations. So we'll start out, let's see. So we'll start out with student project updates. I'll go back over some of the chemotaxis things that we discussed last time. And then we'll do that bacterium macrophage exercise, which we just started. It was a bit in the homework. We just started it in class last time at the very end. I'd like to talk about persistent motion, and that maybe Giuliano will present for us, and wound healing. Those are both exercises that Giuliano developed. So if he's willing to present them, that would be great. And then if we have time, I'd like to start on network modeling. Network modeling is a whole semester in the fall. And so you're getting a very abbreviated form of it, but I will, usually many of the people in this class took the fall class first, but they're not formal prerequisites. You can perfectly well take this class and then the other class. But at some point, it's useful to put the two together. And so I'll give you the absolute minimum you need, I think, to be able to do that effectively, and some tools to learn it on your own if you need it. So why don't we just start with some quick project updates, and maybe start with Connor.
Sure. So Andrew and I met with Roland earlier today. We chatted about some of the design choices in the model that we're trying to replicate. And there were a couple things that design choices that actually were more meaningful and not arbitrary. So I think we're going to pivot and try to copy those. An example of which being the ligand binding sites. The reason they were bit strings was to try to account for the possibility of speciation in later models, which would be nice to have implemented in the base model. I also did what I mentioned I might do last week, which was reprogram what we had in the simulation from the ground up, which actually went really well. It took the majority of my time, some of it behind on other things, but we got the cells stop collapsing to oblivion, which is great. And we also, I got the file down to 2/3 of its original size and rewrote a bunch of the functions. So it's a lot more streamlined now. And CompuCell is generally happier running it than it was with the previous version, which is great. Currently, where we're at is that I'm able to load the cells. I'm able to get chemotaxis going, but they don't seem to be climbing the gradient like they did previously. One possibility is that I did remove the code from that scaled lambda chemo, but the reason I did that is because I don't think it's in the original paper. So I feel like I should be able to get the simulation to work without that because they got it to work without that, presumably. But I'm going to reread the paper again and make sure I just didn't miss it somehow. Besides that, Andrew right now is working on a way to track cell location relative to each other. Because one of the things about the model is that the cells don't actually use up nutrients or eat them. It's just based off of proximity. So one of the things we talked to Roland about is including that and then looking to see whether there's any rotations in the aggregate mass or cells from the middle trying to push upwards as the outside ones become satiated. And that's pretty much where we're at right now. So still debugging stuff, but the previous bugs are now gone, and we also have a slightly more defined direction.
Well, that's great. That bit string can be translated very directly into adhesion using that adhesion flex plugin.
Okay.
Again, the documentation for it is correct, but rather laconic. And so people seem to have trouble reading the documentation and figuring out what they have to do to use that plugin. But in principle, if each bit corresponds to a species, chemical species, you could say that if the two species match, then you stick and you have a negative energy. If they don't, you have either zero or positive energy. And that allows you to pretty flexibly, you have to predefine how many bits your bit string has. That's the one thing you'll have to do. But if you're willing to do that, then you could have, say, 15 bits and have each, if the bits match, then you get a binding and a negative energy. Or if you preferred the other way and you wanted it to be a 0 and a 1 that matched, you could do it that way too. You have to choose. But that's actually pretty well built in. And we didn't really play with that plugin in class. And so we can maybe do that offline. That's the kind of thing where I should probably create some slides to cover it in an exercise. And so maybe working with the two of you separately, we can come up with a little project for that. But I think that's great. And
yeah, that sounds great. Thank you.
Okay, good. Okay, Joel. I guess, yes, okay. So Joel.
Yeah. Can I share the screen? Thank you. So me and Jackson and Dr. Glazier, we had a meeting this weekend, and we go over this paper talking. We were mostly interested in the necroptosis model for this paper, and they actually have two different models. They have a third one that is more complex, but in the sense of what we need to represent for the cells, we decided to go with the simpler ones first, see if everything would work, and then move on to the next one. So we try to do that with these two models present in this paper, done by the supervised by Hoffman and done by his group. And this is I documented everything that we went through because it's an interesting way of looking and analyzing the problems we faced when we were trying to replicate this paper. Following the workflow that we had to read in the beginning of the semester, we started looking at the biological representation of what is happening. And then right after that, the next step would be to look into the math equations that represents the same model. We did that. Here I can make this available if you think it's important, Dr. Glazier. But here I pointed out each function, what they represent on the biological representation, and the same thing with the second one. Then we start, after you do all these things according to the workflow, the next thing is start working on your computational model, right? After you see the biological representations, the mathematical representation, now we try to do that in code. And the next step was for us to choose which tool we're gonna use. And antimony seems to be the best tool for this type of problem that we were facing. And that's why we decided to go with antimony and tellurian to represent this. I left the code here. I can make this available in HTML, which is already everything run. You don't have to run anything, but I can also make the code available if you guys want to play around and change parameters. So it's all comments, so you know exactly what things are doing. For example, this weird thing here is just so we don't see the ugly installing of tellurian here. The model in antimony looks like this. This is the equations for the model A. So we have, if we compare to what was up here, we would see that it's exactly the same equations written in the same way. We decided to use this way of writing this equation, which is writing the prime of the because it was written this way. That's why it's not technically the best way to represent things in this specific problem, but it's a way that works, and we decided to keep as closer to the paper as we could. But some problems start to show up right after that. We thought that something was wrong with our model, our implementation in antimony, because the producing plot was not even present in the paper, which is nothing wrong with that. But if we look, the only plot that they give was the specific molecule PMLKL, which we can see the curve here, but it has nothing to do what we can see in their paper. So we thought, well, we're doomed. Something is wrong with our code. We got back to analyzing the functions again, analyzing the antimony, see if we didn't commit any mistakes, and try to run it again. Another thing that is possible to do with tellurian is to extract exactly the molecule that they were also doing. So we have the exact plot of the PMLKL only, instead of having the total thing that we saw here at the beginning. And as we can still see, there's nothing to do with one or the other, but we knew that the problem was not the equations or our representation on antimony. So the next step is, how can we find a way to figure out what is wrong with the code, right? So we thought that the next logical thing to do here, for this example, we used one value of X. And as we can see on this plot, they were using multiple values or a distribution of X to show the difference of the factor X in this specific model means survivability. It means that it doesn't produce as much PMLK, which every cell that goes above 0.1 is considered dead cell. So the only ones that are alive are below this line, right? So we thought, well, let's do what they did. Let's create a distribution of values for X and see what we can see. And that's what we did. But then the shape of the function, the shape of the PMLKL turns out to be still not the correct, but it shine a little bit of light, because now we can see that is decaying or decreasing. The slope is going negative earlier, depending on the smaller value of X or higher value of X. That gives us an idea that probably something was wrong with the time scale, maybe, because we were seeing the behavior that was represented on the image C much more closer to what we can see at the beginning, at the first 200 units in this example. And so we try to just do 24. Maybe they forgot to multiply by 60. But of course, that doesn't produce the same thing. Then we thought, let's try an arbitrary number. Let's multiply by 10. And we saw that was a little bit too much, because we can start to see that the function here represented the slope starting to going down. And we don't see the same thing in their plots. So Dr. Glazier had the brilliant idea of thinking of another unit of time that they might have multiplied by it, and it was the 7, which is 7 days in a week. Somehow the mistake was, instead of multiplying by 60, they multiply it by 7. To produce the same plot, we had to do this puzzling over the weekend, trying to find exactly. And we can see now that they have almost the same representation. We can see almost the same shape. The only difference was that we were using an uniform distribution. And in their case, they claim to use a log normal distribution. So we tried the log normal distribution that they wrote in the paper, and turns out that that is not the same that we can get, right? As you guys can see, is very much steep at the beginning, which has nothing to do what we can see in the image C. So now it's the turn to fiddle with the log distribution and see if we can find something that is similar. One of the first things we do, let me see what we did different here. Oh, we just cut off this multiplier by 0.05, since we see a lot of the values are stuck into this side of the distribution. So maybe if we take that down, we can see more of a distribution. And yes, we can start to see that the values are more distributed along the whole area. But it's not perfect. We don't see the same thing. Then comes another time-consuming way of figuring out what was the numbers that they used to find something similar. I don't know how to explain to you. It was just a try and error over and over again. But this is something that is the closest that I got. It has nothing that I can reason. It's just trying to control the mean and the max of this distribution and trying to fit in the same pattern that we were seeing on their paper. This is the best that I got, but it will become clear on this next cell that this is not good enough, because we have a survivability here of 30% still, because the density down here is still higher compared to their paper. And their paper just show 6% of survivability in the same scenario. But in the interest of time, we decided to move on and try the other model, because it was a different distribution, and that is a little bit more promising, although it's still wrong. The one they used was a normal distribution here multiplied by 0.5 and a value between 7 and 8, and another sum up with another normal distribution with the values between 14 and 2. I think, yes, they say that that's the mean and the variance of the distribution. That doesn't produce the same plot as expected. So the one that we find out that is closer to what we can see there is something like this, that is also completely arbitrary. This value of 14.7 is completely arbitrary. That generates something similar and the survivability is closer. They are 18, ours is 19. We can blame that on the random of the distribution. So that's my report for this week. And if you guys want to see a little bit more of this whole process, I still want to finish and put more information here. But this is as much as I could gather on our time frame.
Great. Jackson, did you want to add anything to that presentation?
Not the presentation, but I did try to fiddle around a little more with the distribution to try to find the graph they gave us. And I spent, I think, like an hour on just different numbers, just putting in different numbers. And none of them showed what they had. So,
Well, it just shows that you could be a very famous professor and publish in a fancy journal and it could still be wrong. And so this is something, I mean, this is something that I was talking about with Joel and Jackson, which is we have a friend in England, Rahman Sharif at the EMBL, and his specialty is model replication and reproducibility. And so they did a study where they took 500 papers of this kind of model, where the code for the simulation was published. So in principle, you'd say, oh, it's easy. You download the code and you run it. And then they asked the question, could you replicate the results in the paper? And the answer was they could do it 1/3 of the time. So then for the remaining 2/3, they wrote to the authors of the papers and said, because the papers are always supposed to say, if you need help with this, reach out to me. So they wrote to the authors of the paper. Half the people never responded at all. Of the half that did, who helped them, only 1/4 were able to replicate their own work. So I'm not saying that you shouldn't be careful when you are fail. Clearly, if your code doesn't run, there's a mistake. And it may well be that when you're learning, the probability of the mistake being yours rather than in the paper is higher. But you need to learn to be skeptical even of published results by big names in big journals. And so just because some paper says that something happened in a simulation, that doesn't necessarily mean that it did. And I think this is a very good example. The simulation seems to run. It does seem to do what they say it does. But because the axis labeling somehow was wrong, trying to interpret what they did is quite hard. And because they don't give the code that took the simulation and turned it into the figures, you have no way of knowing what they did. And that's really tough. Now, in principle, there should be MATLAB code. And if you downloaded the MATLAB code and walked their code, provided it worked, you might be able to find out what they really did. But it's not easy. And so it's something that you need to be aware of. You need to learn to be willing to challenge published results, not to take them on faith. And so I thought Joel and Jackson, thank you for doing that. I know it was a lot of work to do that presentation, but I hope it's interesting for everybody to see that you can run into this kind of problem, and also how you can start to deal with it. And even if you're not able to deal with it completely, at least you have some idea that you have the right math, that you figured out some of the mistakes, and then you can move forward from there. So thank you both for doing that. That was great. Alex, how are things going for you?
Fine. I don't have any updates really. I met with TJ last week. He was happy with the progress I'd gotten. So I didn't do much work the past week, but other than that, for the future, I'll be working on seeing what, if any, GPU-based acceleration can be done for the rendering pipeline.
That would be great if we can get things on GPUs. That would be fantastic. How that will play with Zoom, which seems to be its GPU hog, is another question. I'm always having to go in and either turn GPU assistance on or off with Zoom. Every time they seem to release a new version, it crashes. And so, but I guess it must be difficult to write a software package like Zoom that has to run on every computer.
Yeah, it's the cross-platform thing is a lot of work. Thankfully, for GPUs, there's OpenGL, which is a library pretty much all of them support. It's like it's not a driver, but just lets you an API essentially. But so I'll be looking into two things there, because you use VTK for rendering inside of CompuCell. So that offers in itself like GPU rendering classes. I'm not sure if those actually get used already, like for free. They might actually be. So looking into leveraging that if it's doable, and then just the feature extraction bit as well. But then keeping track of, because data movement is a big problem now, because if you pull your data from the feature extraction, which is on the CPU memory, copy it to the GPU to then extract, copy it back, then copy it back again to do the rendering, it's kind of a waste of everyone's time. So not doing that is a requirement.
So you have to be smart about your data flow. I guess some of the new GPUs, they have these big stacks of GPUs, and then the inter card communication is very fast, but you can't count on that in somebody's random laptop.
Yeah, I mean, most people don't have multiple GPUs with that SLI interconnect. Just one is usually hard enough to find nowadays.
Well, maybe not this semester, but going forward from this, TJ does have connections and some connections at Kitware. So in principle, if there are issues, we can go to them and ask them for some help.
Yeah, I guess, so at least that the experiments we've been doing this semester, they really aren't large enough to need multiple GPUs. Because modern GPUs, even if you get like an inexpensive one, you're looking at 4 more gigabytes of on-device RAM, which is more than enough to hold these small simulations.
Yeah, typically in class, of course, we'll do small simulations because we want it to run in a few minutes. For the production simulations that Giuliano or TJ do, well, most of the ones we've done with the immune system are sort of 2.5D. They're still fairly small. Although TJ's flu simulation, I think, takes about 10 hours to run. But when you start going to things like cancer simulations, which really have to be done in 3D, on 1,000 by 1,000 by 1,000 lattice, you want to run 100,000 Monte Carlo steps. That's 100,000, right? 10 to the 4th, 10 to the 5th, times 10 cubed times 10 cubed times 10 cubed, 10 to the 9th, and the 5th it's 10 to the 14th steps. And that's manageable these days. That's not crazy, but it's a lot of time steps. And so you do need you want to be as fast as possible if you're going to chew through 10 to the 14th steps. So that's great. Okay. Well, thank you for the update. And I'm grateful to you for working on that great problem. Okay. So Josh, I guess you're I don't mean to always pick you last. I just go by the order on the screen, but next time if we're the same screen order, I'll reverse it so you don't have to be last. Josh, what's up?
Oh, it's fine. I don't really have much to report. All I have to do is add a steering panel, and then I'll upload it to NanoHub, and then I just need to work on the documentation and the paper and stuff.
That's great. So I was hoping to do some slides on steering panel this week, but I'm not going to get to them this week. Giuliano, maybe you could present some basics on steering panel for next week.
Yep.
I think at the moment we have one slide that basically just shows the code, but there isn't an exercise for it, and there isn't really a lot of supporting material. So we need to do a better slide deck for.
Well, there's a documentation page that I found that's pretty helpful. I'll just use that.
Great. Well, I mean, you're very independent, but I still, for everybody, it would be great to. I'd like to show that material. We could just take, of course, start with the documentation, put it on slides. That may be a way to start. But one of the things that we face with, actually, it's not a problem. You're not going to say something, and then it's because I was just looking at Joel with tellurium. We're not going to face that problem with the CompuCell steering panel. One thing you face with the Jupyter notebooks on NanoHub is that they have it often use old libraries, I mean, really old libraries. And so the steering panel options, the GUI options in the tellurium interface that Joel was just showing, use a package called IPyWidgets. And a lot of the IPyWidgets aren't implemented in the NanoHub version. And so that can be an annoyance because you do a beautiful Jupyter notebook with all these nice GUI, and then you upload it to NanoHub, and it says, you know, IPyWidget blah 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 not found, or not, so give you some permission error, and so then you have to redo it with a different widget. But we should be okay on that. One thing we're hoping to do in CompuCell is to actually, one, get it into Jupyter notebooks, and two, to add a few extra widgets for user interface. That one steering panel app we have it's not bad, but it's really not enough. There should be a little bit more flexibility. And that shouldn't be that hard to implement computation. It's one of these things that takes, implementing it is not that big, but documenting it takes a lot of time, and making sure that it works under all circumstances takes a lot of time. So those are the two problems. Okay, well, thank you, everybody. That was great for the updates. Okay, so just for review, last time we did the self-constructed gradient example. That's an example that I really love, and I hope people maybe play with it a little bit just to see how that works. I think there's a lot to explore there, and I really would like to, somebody wants some extra credit and wants to write an exercise. I'd be happy to give somebody some extra homework credit for writing a good exercise for that, because I think that's a really pretty simulation, and it shows up in biology a lot, both at the level of cells in your body, at the level of colonial organisms like bacteria in the environment, at the level of animals in ecosystems as well. And then we looked at changing the chemical field data for chemotaxis. The one thing that we found was that it's not an attribute of the cell directly. It's not like cell.volume, cell.target volume. And the reason for that is that it's a pairing, and this is always a problem when you're dealing with objects that are not, or relationships that are not intrinsic properties of a single object, where they have two or more arguments. And so here, the chemotaxis data relates a chemical field to a cell. And so we have to then call this chemotaxis plugin, add chemotaxis data, or here, get chemotaxis data, to establish the relationship between that particular cell and that particular field. There, in the add chemotaxis data and the set values, there are rather a lot of options. It's not just lambda that you can change. but those are more subtle, and if you're interested, you can look them up in the manuals. Again, one could imagine a lot of other kind of chemotaxis formalisms. Over the years, people have developed a lot of different chemotaxis algorithms because the biology is different. In our case, all the surfaces of the cell respond to the chemical in more or less the same way. And we've got the ability to say that surfaces in contact with another cell of a particular kind can respond differently. And that's something. But in reality, the front and the back of a cell, when the cell is crawling, respond to the chemical differently. The sides of the cell respond differently. The cell, when it's extending and when it's retracting, are different. And so there are a whole set of mathematical architectures that people have developed to try to deal with those variabilities. And CompuCell implements a couple of them, but not by any means all of the ones that have been used. And so it might be of interest at some point to add those. But the key thing is that once you have this handle to the chemotaxis, then you can use it like any other object and it has its attributes. One of the annoyances in NanoHub CompuCell is that print doesn't work. So that's something we need to fix in our next release on NanoHub. So my favorite thing, which is to dir an object to see what you can do to it, doesn't really work very well. You can save it to a file. You could dir it and then save it to a file on NanoHub, but you can't print it to the file. And then we, in a sense, skipped over this exercise, deciding that it was partly in the homework, which was to look at chemotaxis speed as a function of parameters. In this case, in this simulation, we were scanning the lambda chemo, but it's also quite informative to scan cell volume, which would be related to Connor's and Andrew's project, or to scan something like lambda volume. When you make lambda volume bigger, the cells become stiffer, they don't move as easily. And so making lambda volume bigger, you'll see the cell will move more slowly for everything else being the same. Or what if you change a parameter that doesn't have an obvious effect, like the contact energy with the medium? Does that speed it up, slow it down? What does it do? And so being able to scan all those parameters and see what happens is sort of interesting. In this case, we had the cell essentially bounce back and forth and measured its velocity. Plotted, in this case, the velocity in voxels per Monte Carlo step versus lambda. And what we saw, this is on a log scale on the x-axis. What we saw was that there's this region where the increase in velocity is fairly slow. Then there's a fairly big region where you have essentially a linear relationship, and then you saturate. And you see that no matter how big the lambda was, you couldn't go much bigger than 0.2 voxels per Monte Carlo step. And this does reflect a fundamental speed limit in the methodologies that we use. If you push things too hard, you'll see cells break apart, you'll see cells disappear. We've seen that in the simulations we've done. And so, again, this is a seemingly very, since maybe a dull problem. But when you dig into it, there's a lot to learn from. And then the last thing that we did was talk about logarithmic saturation of chemotaxis and tried out putting in an external function where lambda chemo, we would by hand rescale it by the concentration. Here I use the center of mass concentration. Giuliano made a point that we could use this function secretor amount seen by cell to get a more accurate estimate of the concentration. It would be sort of interesting to compare the results. I use center of mass mainly because it's fast. Pulling the as seen by cell is a little bit slower. But if you're only doing it once per cell per Monte Carlo step, it may not matter very much how long it takes. That may be a small part of your computational burden. But if you have an exponential chemical field, what you see when you put a cell in that field, in this case, we're starting at low concentration, it's moving up the gradient. The cell moves very, very slowly, and then it accelerates without rescaling. If I do the logarithmic rescaling, the cell moves with essentially a constant velocity throughout that gradient, which is really nice because otherwise you spend the cell sits here and does nothing for forever, and then it comes over here and it falls apart. And so that rescaling really is a useful trick to have. The one thing you might have to play with at small concentrations, very small concentrations, is this value of 1 here. There has to be some number, because if you use 0, lambda over c could be lambda over 0. You don't want to divide by 0. But the bigger that 1 is, essentially this is controlling, saying that for small concentrations, it's really not rescaled. And so you need to set that scale. But this works pretty well. And again, there's a lot one could play with in this. And it's not unreasonable in the sense that if you look at cells crawling in chemical fields, very often their velocity is independent of the concentration. They either move or they don't, but if they're moving, they move at a fixed speed. For very small concentrations, very small gradients, you do see some acceleration with great steeper gradient, but most of the time cells, once they start crawling, they crawl forward at their speed. So that's something to think about. All right, so I wanted to show you just a few things that we could ask, some questions, generic questions of the kind Joel was talking about in sort of the thinking about problems. I think I went over this a little bit before, but I want to come back to them now that you can do chemical fields, you can do chemotaxis. We have the idea of cells absorbing, we've worked on. And so if you have a diffusing chemical and cells are responding to it, here are some of the kinds of questions you might ask biologically or biophysically, which will determine what you're going to do in your simulation. And some of these are pretty simple. Some of them may be more subtle than they look. To begin with, where is the signal? Where is the molecule? Is it everywhere in space? Is it under the cell? Is it excluded by the cell? Where is it? In a lot of simulations, using the Potts methodology that we use, the assumption is that the chemical is everywhere and that the cell doesn't have any effect on its concentration. And that's clearly not true in reality. Cells obstruct the flow of chemicals. And it's a little bit surprising, perhaps, that those simulations often work pretty well. Even if the chemical is confined outside of the cell, you can ask the question, is it freely diffusing or not? Because the inside of cells is full of proteins and other and membranes. The outside of multicellular environment in your tissues is full of extracellular matrix, which are fibers. And it's not like diffusing in water. Again, we often ignore that, but we'll find that many chemicals signals, and this is our self-constructing gradient example, bind to the extracellular matrix pretty tightly, in which case they either don't diffuse at all or diffuse very slowly. And once we've just got the chemical available to our cell, we have to ask the question, does it cross the cell membrane? Is it excluded by the cell membrane? How does it diffuse? Does it diffuse at the same rate inside the cell and outside the cell? Often these are second order questions, but in some simulations, they wind up being very important. It's definitely important to know whether the molecule is everywhere or whether it's confined to some substrate. So if you have a cell crawling on a substrate and the chemical is in the substrate, that's very different from a cell that's experiencing the chemical all the way around it in 3 dimensions. And that typically makes a big difference. In principle, if the cell excludes the chemical and the cell moves, it causes flow of the extracellular environment. We tend to neglect that, but we should be asking the question, if we're neglecting it, is it we're neglecting something that's important or not? And typically, if the flow velocities and cell movement velocities are slow compared to the diffusion times, then it's okay to assume that there's no advection, no carrying. If the cell movement is fast, then you can't get away with that. And then when a cell receives a chemical signal, does it pull it out of the environment or not? Usually it does. Usually sensing is destructive by cells. But typically we assume it isn't. If it is destructive, are we eliminating a 1 in a million part, in which case we can ignore it, or are we removing 10% or 50% of it when we sense it? That probably depends on the concentration of it in the environment. Once we've found a signal, and this will come back a little bit to what Joel was talking about, and also to what Connor's working on, how does the cell receive the signal? Are there receptors on the cell surface? Are they everywhere? Are they only in some places? Does the receptor only function when the cell's in contact with some other cell type? And then what's being sensed? In our simple chemotaxis simulations, we're typically sensing the concentration of the chemical. But you could imagine that the cell detects the direction of maximum concentration, the direction of maximum gradient, direction of minimum concentration. All of those are rather different. And as an example of this that I think I may have mentioned before, if I hadn't, I'll say it again. It's worth saying again, even if I did, which is, as you know, when ants move, they lay down pheromones. And so you say, okay, the ant follows the pheromone trail. But if you ask the question, if I'm an ant and I'm laying down a trail of chemical, and I say I'm going to go up or down a gradient of chemical, where's the steepest gradient? What direction is the steepest gradient with respect to the trail? Is the question clear? Suppose I'm an ant and I crawl from this point to this point, from here to here. So I grab it like this. I lay down a chemical. And now I ask you the question, which direction is the gradient of the chemical steep?
There will be less where it started, right? That's the.
Well, presumably if the chemical doesn't last forever, then where I started, it's older, and therefore there'll be a little bit less. So the concentration will be a little bit higher at my current position than it was at the origin. In which case, if I want to find my way back to the origin, I have to go down the gradient rather than up the gradient. But the ant doesn't live in one dimension, it lives in at least two. And if I look from here to here, suppose this is 10 meters apart, the concentration goes from not zero to whatever the maximum is over 10 meters. What if I look perpendicular to the trail? What's the gradient? If the ant was never here, here's my trail. If the ant was never up here, what's the concentration?
Lower up above.
Zero.
Yeah.
On the trail, the concentration is large. And so the gradient perpendicular to the trail is very, very steep. So if I follow the gradient of the trail, it's completely swamped by the perpendicular gradient. and your ant will never find going up and down gradients. Your ant will never get back to its home. You have to have a different rule where the ant looks for the largest concentration or the smallest concentration, where the ant actually looks at the difference between the tail and the head end and makes the choice. Just doing a gradient descent doesn't work. And it's actually rather tricky to come up with an algorithm that allows the ant to find its way home. So that's why I wanted to give that example, because people say, oh, the pheromone attracts the ant. Well, if that's all that happened, ants would get lost all the time. It's a more complicated rule. Okay. And then we already talked a little bit about saturation or adaptation. Is how does the number of receptors change in response to the signal level? And then sometimes the signal is the receptors themselves are saturated. Sometimes they're released, sometimes they aren't. And then once the signal is received, basically, how does the cell respond to that signal? There are cases where a chemical signal is received and the cell does something continuously, like chemotaxis. There are other cases where there's some threshold, there's an integrate and fire like a neuron. Once the cell's done its thing, there's some long period of what's called a refractory period where the cell can't respond again. And then there's some additional questions if you're talking about migration. What does the cell do if there is no chemical field? CompuCell cells diffuse randomly in space, whether there's a chemical field or not. Is there a minimum concentration which you need to detect? So Connor's working on the problem where that was discussed. Does the cell respond as a unit, or do individual patches of the cell respond separately? We gave the example of neuronal outgrowth, where each little patch of the membrane behaves completely independently of every other patch. And then things like the force velocity relationship or the signal velocity relationship, which is what we analyzed in that exercise. And so there are all of these questions. Does the cell integrate multiple signals? What if I'm trying to go up one gradient and down another gradient at the same time? What do I do if those signals conflict? And generally, we respond to multiple signals simultaneously. You want to go north, but you smell something good coming out of the bakery, so you walk into the bakery. Those kind of things. And also, is the motion continuous or pulsatile? Many cells will move. They'll move forward, they'll stop, they'll move forward again, and they'll stop. And that pulsatile motion can actually have very different physics in its results. In embryonic development, a contraction in particular tends to be pulsatile, and that winds up having very different long-term properties in terms of the way this tissue reorganizes from continuous motion. So those are some questions that you might, as I say, I mentioned a little bit of this earlier, but now that you've been working with these ideas, I hope some of those questions make more sense. So let's come back to this version of the homework problem that we ended with last class. And I'd like to do this as an exercise a bit together. Whether we do the whole exercise or not is another question. But I told you about this beautiful old movie from the 50s of the macrophage chasing and eating a bacterium. This was the results of a simulation of that, which was the final result of one version of the exercise that we want to do. And we have this basic idea that the bacterium is secreting a chemical. The chemical diffuses, and the macrophage moves up the chemical concentration gradient. Wow. I had no idea that there was sound associated with that movie. That was reasonably embarrassing. So there's our movie again. It's a better video, although, which I guess is why I picked it. But if I'd known it had sound associated with it, I wouldn't have done that. And so if we watch this movie a couple of times, we have to think about what it's telling us. This is thinking about the kind of biological model or qualitative model that we're talking about. So the first thing we see is the bacterium, or actually a couple of bacteria, are small and they're compact. So treating them as smaller quasi-point-like objects is reasonable. It's actually not clear from this movie if the bacterium is moving actively or not. It certainly looks like the bacterium is fleeing in front of the macrophage. Here I said neutrophil, so inconsistent in my identity of the cell. But in fact, it's possible that all that's happening is that the fluid in front of the moving cell is being pushed, and you're basically advecting the bacterium away. Now, since we don't have a concept of advection in CompuCell, if we were trying to implement that, we'd have to cheat. You can actually make the medium into a cell type, basically divided into lots of fluid particles, and it works, but it's a very computationally expensive thing to do, so we tend to avoid it. If somebody wants to learn how to do that, I can show them, but it's not much fun to have to do it. The neutrophil has a fairly oval shape, which means its surface to volume area is pretty big. It seems to be able to see the bacterium from a cell diameter away, which means that the chemical that it's using has to have a fairly long diffusion length. It definitely seems to move in the direction of the bacterium directly, although it seems to ignore this one, right? It moves and chases that one, but it doesn't seem to pay any attention to that one over there, which is going to be inconsistent with the result, what we're going to do. Maybe those bacteria are dead and not secreting the chemical. Possible, I don't know. It does eat the bacterium eventually. If we actually track the velocity of that cell, it's relatively, the velocity, of course, I'm using velocity to mean speed. The speed of the cell is relatively constant. Velocity, of course, is changing all the time because the direction is changing, but speed is relatively constant. There also are these erythrocytes, red blood cells in the environment. They're pretty round, although some of them are spiky, which means their surface to volume area is fairly small. They don't seem to be doing anything active. They're certainly not crawling. And we happen to know biologically that erythrocytes are essentially dead cells. Dead is maybe the wrong word. They're passive. They have no nuclei, and they don't have a lot of active motility. They can change shape as osmotic pressure, pH, and salt and other things change. But they're essentially more like lipid vesicles than anything else. And they don't do anything active. None of the cells stick to each other. And one thing that we can't really tell, although maybe if we watch the movie enough we could, is whether whatever that signal that the bacterium is sending is diffusing through the erythrocytes or not. In the homework problem, we assumed that those walls blocked the chemical, but we could have let the chemical go everywhere and asked the question, how is it different? And that would be sort of an interesting thing to look at. Sorry about the noise. I'm not sure why it's so fast. That's also strange. I guess what they did here in this version, they probably took a version of the video with the commentary, sped it up, and then didn't notice that they had forgotten to delete the audio track. Next time I talk to Phillips, and I can tell him he should update the video for his textbook. I'm a little intimidated. He's a Caltech. He's a truly great scientist. I don't think I'd ever dare to say something like that to him. I should. Okay. So our hypothesis is going to be that the neutrophil is chemotaxing towards the chemical secreting by the bacterium. We're going to have 3 cell types: bacterium, neutrophil, erythrocyte. We're going to have 1 chemical field to begin with, which is bacterial chemoattractant. And we'll start out with 1 bacterium, 1 neutrophil, and multiple erythrocytes. Boy, that's annoying. And it didn't play when I did this. I don't understand, because when I did this check this presentation on PowerPoint without playing it for you, it didn't have the audio. So there's something odd. I think this is maybe a Zoom PowerPoint interaction. Not sure where that's going. And so then we're going to start building this up. We're going to say our bacterium has volume, surface area, potentially some kind of motility, although probably not. Bacterium doesn't seem to do any active crawling. It has to have some kind of contact energy, because if it doesn't have any contact energy, they'll stick. The neutrophil has a volume, surface area, definitely is motile, doesn't stick to things. The erythrocytes have volume, surface area, they stick, they're not motile. And the chemical, if we wanted to send a signal, has to diffuse and decay. Bacterium is going to be able to secrete the chemoattractant. Neutrophil will chemotax in response to the chemoattractant. We don't know if the neutrophil absorbs the chemoattractant or not. That was one of our questions. And we don't know whether the chemoattractant diffuses everywhere or just in the liquid around the cells. And so we're going to have to specify things like what's the diffusion constant, what's the decay rate, and that will give us the diffusion length. So we're going to have to make sure our diffusion length is reasonable. We're going to have to say how fast this bacterium secretes the chemical. Is it a constant rate, constant level? How strong the neutrophil chemotaxis is? Do we have saturated chemotaxis or not? If the neutrophil does absorb the chemoattractant, we have to know how much of it takes out, or how it saturates. If the chemoattractant doesn't diffuse in our red blood cells, we have to specify that it doesn't, because by default it diffuses everywhere. And we're going to have to specify contact energy so everything doesn't stick. Now, if we're going to want the bacterium to get eaten at the end, to have the bacterium eaten, we're going to have to make it stick to the neutrophil. So there's going to have to be some rule that says under certain conditions, the adhesion changes. And because we can't have it not stick for a long time and then start sticking, something has to change. And so that's something we'd have to think about. Okay. So if you want, yes, let's do this as a little exercise. You could start with your homework exercise if you like. There's also a version of the simulation in the demos. The version of the simulation of the demos is not the most pretty in the world. And Giuliano, could you put the specific pathway to it? Because there are I think a couple of versions of that simulation in the demos. And I thought I had put that in the slide, and I apologize. It's in one slide, but it's not in the one that you need it in. And why don't you try seeing what you can do? You start with a single bacterium and play with the diffusion constant and decay constant, the chemical field, and adjust them until the neutrophil finds and follows the bacterium. If the diffusion constant is, if the diffusion length is too short, then unless the neutrophil is right up against the bacterium, it's never going to find it. If the diffusion length is too long, the gradient's too shallow and the neutrophil won't really move much in response to the gradient and to the bacterium. And if you have the problem that we faced before, which is when the neutrophil is far away, it doesn't move, just sits there. And when the neutrophil gets too close, it gets distorted because of the gradient being too steep, then try the saturation that we just have gone over and see if that works. So is that okay as an exercise to do in class? Does everybody have that? Does everybody have the material that you need to do that, either your own homework problem or using the version that's online in the demos file? Juliana, were you able to find the hierarchy just so we can make sure everybody finds the demo properly?
Trying to. I could find it in 2 seconds the other day. Not so much today.
Why don't people take a look on their own? If you can't find it, come back and I'll mute myself and look for it while you guys are working on it. Are there any questions?
Do you recall the specific name of the file?
Oh, so Giuliano suggests that there's of which file?
Of the one that you wanted us to look for in the demos I was looking for.
So Giuliano suggests using demos, bacterium, macrophage, new style.
Yep. The only thing about that one is that there is a if MCS equals 300 stop simulation. So comment that out.
Oh, okay. So we're using the demos that are on CC3D. Okay. Got it. I thought it was on the material from the student material. Never mind.
I think I saved a version there too, but no, these are actually the packaged demos. And it's not the greatest demo. But the idea was it would start with one that wasn't great and then you'd gradually modify it to make it more realistic. And if anybody is not able to find it or needs help, Giuliano and I are here to help out. Oh, one thing I strongly recommend, if you use the demo that's packaged with CompuCell, do a save as to your desktop or someplace else, because it will happily overwrite the demo. Twitter will happily overwrite the demo, and then you won't have that demo in its original form anymore. Those are not write protected. So load it and then do a save as to someplace where you have a working copy. How's it going for people? That Bacterium macrophage new style one has some code in it that's not very helpful. And so you might want to try that alternative, which I put in the chat. So, but either one. So was everybody able to find the demo simulation?
Yeah, I did find the periodic one.
There's the one that's the periodic one is one possibility, and then the one that we recommend that Juliano recommended, that new style one, is also fine. It just puts up a plot of something that's maybe not so exciting.
Yeah, I started with the new style one, and I think I have it set up right now. I'm just scaling lambda chemo so it doesn't charge towards it at rapid speeds as soon as it sees it.
Yeah, putting in that chemical saturation helps a lot. And for the person who's already done with the first part of it, I can look ahead to the next slide, which is to make it a little bit more realistic. So my next exercise for you would be to try using 2 or 3 bacteria and see how the neutrophil responds when it has to choose between multiple targets. And then when you're done with that, you could have multiple neutrophils. If you're really done with that, then you could ask the question, what if the neutrophil absorbs the chemical field while detecting it? Does that make a difference? But I want to give people time to do the basic exercise, but for whoever got ahead, you can try that. I'll leave this slide up for a second while people are working on the first part.
So I have a quick question. Maybe I'm
Sure.
just forgetting.
Sure.
So usually when we are looking to manually tune lambda chemo, it involves commenting out the center of the like the definition from the XML file. But that's also where we get to select if we want what type of chemotaxis we want, right? Like whether it's the saturation or linear.
Well, so you could do it there, or you can do the saturation by hand, where you keep rechanging lambda chemo by
Okay.
hand in the step function.
Okay. Okay.
They're both, I mean, they're both legitimate. So, but I would recommend doing the saturation in the, use the regular linear chemotaxis, but adjust the lambda chemo value by measuring the center of mass concentration and then rescaling.
Okay. Thank you.
For the people who've gotten the basics, are you able to work your way down the list of improvements a bit? We don't have to get through all of it in class, but I'd like to make sure everybody has the basics down. And then we can explore more in homework. Would anyone like to show what they've got so far? For the people who are finished, at least partly finished?
Sure, I can show it. Just give me a second. I'm trying to change something. Just see how it works. Just give me a minute.
Again, to work your way through the whole stack of exercises takes quite a while. But I hope everybody gets at least the basics. Was everybody able to at least load the demo and get it to work?
I loaded the demo, got it partway to work, started experimenting, broke it, and I'm now trying to figure out how I broke it. So
Well, that's one reason not to overwrite the demo file.
I
That
made
way, at
sure
least
to
you could
save
go
it.
back to the back to the original.
Yeah, I can show you something that I
Okay.
changed and see how it looks. So, okay. The changes that I did was now the macrophages actually also devour the diffusion. Beforehand, the diffusion would be expanding all over. And now just the fact that they're also eating the secreting field. I think was part of the exercise to see if it would change something. But yeah, see, they're still divided. It doesn't know if you want to go for this one or that one. I didn't do that exercise still. But yeah, I'll keep working on it.
Do you have saturation in that one or not?
I do. Well, I think I do. Let me see. Let me show
Are you adjusting lambda chemo
Yep.
by the concentration?
Yep. Yeah. I use the example that are on the web page on the documents there.
And why don't you show it again? I have one other question about it.
Sure, let me share my whole screen so I can share the code too. Okay, see if I can open the code here. Okay. No, this is in the way. Okay. I have limited space now. Anyway. Yeah, the way that I implemented it's right here. I use the diffusion to be bigger just because I wanted to start it earlier. This file also have a limit of steps. If someone is having trouble with that at the very beginning here, you can change the number of steps. It is like around 10,000. It's not enough to see a lot of things happening. I
Okay,
don't know.
so I
Go
have a
ahead.
question with the one you just showed with the lambda chemo adjustment. Let's look in there. Python.
Yep.
So you're not rescaling it by the concentration.
No, I used it the thing that I found in the manual, which is not the same as I think on the slides, right? Because I think it's you're using the log adjustment on your slide. Yeah, here is not the log adjustment.
So what will that do? That will continually decreasing lambda chemo.
Yeah, I think as close as it gets to the cell, right? As close as it gets to the bacteria.
Well, so let's see. It goes over the cells. If it's a macrophage, it pulls the chemotaxis data, and then it sets the lambda chemo to be the current value -3, and then resets it. So lambda chemo will go negative and then get to be infinitely negative.
It is true. So we should see it getting away from the.
No, because negative is going to attract more.
Oh, okay.
So
Okay, so I have
basically, what you're doing is you're making it
make it
respond more and more strongly.
He wants that bacteria. What can I say? Yeah, I think I got it from here. From the.
Right.
Yeah.
Right, but that's not.
Yeah,
that's
it's not what the exercise.
I mean, it's perfectly. I mean, it's perfectly good Python, but I don't know what it's.
Yeah, I just couldn't remember the
syntax. I should probably have had.
Yeah, I couldn't remember, so I was like, I don't know. Let me see if I can find it somewhere.
So why don't you try reading the chemical field and rescaling by the chemical field?
Okay.
So what I would do for that is I would save the initial value of lambda chemo somewhere, and then have that available and keep multiplying it by 1/1 plus the chemical concentration at the center of mass of the cell.
Okay.
Forgot how horribly Zoom makes code look, so feel free to just ignore that entirely.
Yeah, looking in the chat, using amount seen by cell is a good way to do it. Probably better than center of mass.
The problem that I'm trying to solve now is how do I make a cell focus in one scene bacteria instead of like chasing the nearest secretor, or I'm trying to solve that problem in my head first, but it's hard to think in terms of the CompuCell logic.
Well, I think it's not. I think that probably it's not possible using. This was one of the examples where I said that chemotaxis could be complicated. So in reality, when the cell has a leading edge, the leading edge will respond to the chemical, but the trailing edge won't. But we don't have any way of doing that effectively here. If you used a compartmental cell, where only a part of the cell surface responded to the chemical field, then it would be able to do that, because then only the part that was closest to the bacterium would respond. The rest of the cell wouldn't care.
It did seem that setting up the decay for the macrophages helped in the sense of like following this target more than when it's not having decay of the secretion. But I go back to the problem of like whenever it has chew, it doesn't respond well.
Yeah, that would be what I would expect. I think actually getting it to follow only one and stay with it is going to be difficult. But that's the kind of thing where if one were really going to explore this in a serious way, that you'd have to think about that carefully. I'm not here for. We've spent a lot of time on the exercise, so I don't want to spend too much more time on it. But that's an excellent question. Does anyone else have something they want to show for this? I updated the slide to give a little bit more specificity based on your comment. Again, using the center of mass, it's fine to use the total amount seen. That's probably actually better than center of mass. That exercise was written before you had the amount seen as an available quantity. How are people? Go ahead.
I do have a question. Whenever you make a cell frozen, it just stay as a square, or there is a way to establish it as like a rounded shape?
Well, you have, if you use the initializers only know how to draw squares. So you're stuck.
Okay.
You can make it, there are a number of ways you could do this. One way would be to make the cells initially of not create two cell types. It's pretty ugly, but you could do it. Create a cell type which is a frozen cell type and another cell type which is a non-frozen cell type.
Mm-hmm.
Let the cells move around for 200 Monte Carlo steps, and at Monte Carlo step 200, change the cell type to frozen.
I see.
That would work. Another way would be to use this not very elegant PIF initialization and read in the pattern. The best way to do that would be to write out, to do what I just said, which would be to let the cells relax, write them out, and then read them back and say they're a frozen cell type. You could draw them with Python, draw circles initially of a frozen cell type. But that's a limitation on the initial condition generator. So.
My intuition would say also that if we make it frozen, it will not move when the macrophages try to push its way to follow the other one, right?
Yeah,
that's right.
Gets, okay, everything gets
stuck.
You definitely don't want the red cells to be frozen.
Yep.
To replicate the movie.
But another way to have circular frozen cells is to have a list of pixels that you know form a circle or close enough to a circle, and plop well, 10 of those for a cell.
Is it possible, for example, to change the temperature of a single cell in the sense of like it's not trying to make so many copies, so it's not moving so much around?
It is possible to do it on a cell type basis. I'm not sure about for individual cells.
Okay.
I would recommend using lambda volume. If you make lambda volume very large, the cell freezes.
Mhm. Okay, I'll try something.
Other questions about this?
So I'm not at this point yet, but if I wanted to get it so it was actually eating the cells, would the best way to represent that just be that if there's contact, like if there's a neighbor of the other type, to set the lambda, like to set the target volume to 0 for the bacteria. So just kind of, I feel like that's the easiest way to represent death, but I'm not sure if that's true.
That would work. The other thing to do would be to overwrite the voxels, basically force the cell to become part of your cell. There's a function in CompuCell that does that, although it's not very pretty. What you might want to think about, and this is a case where 2D versus 3D is a little bit misleading. In 2D, the ratio of the volumes is already large. In 3D, it's even more diverse. But in principle, you could take the target volume of the bacterium and transfer it to the target volume of the neutrophil, since the material target volume in some way represents the mass of the cell. And so you can do that. When we did this, when we were working with Josh Laksina at NIH, where he was internalizing his macrophages and neutrophils were eating eukaryotic parasites. Those are a little bit bigger, and so we wanted to keep track of the volumes. And so when a cell ate another cell, it first we had it attached, so we had them stick, and then we'd actually transfer the volume, target volume, from one cell to the other. So that's a good question. Okay. Anything else people want to ask? Maybe we've had enough time on this exercise. Why don't we're already at 7 o'clock? Goodness. Why don't we take a 5-minute break, and we'll come back and move on. Giuliano, maybe you can type this up as a little homework assignment, so that people who didn't finish it get to work on it. If there are any questions, Josh and Jackson, if you have any questions or stuck on any of this, please feel free to ask. So I don't want to move on and have people having the basic part of it. Again, it seems like a simple thing to state, but in fact, getting it to work well requires quite a bit of thinking. Okay, so I'll see people back at 7:05. Is that okay? And then we'll move on to our next exam. Okay, are people back? Everybody back, all right. So I'd like to go on to another problem, which maybe we should have done, thinking about this, Giuliano, next year, we should have done this before rather than after chemotaxis, which is persistent cell movement. And in this case, we're going to model the way cells crawl independently of an external chemical field. And again, the thing to remember, and we talked about this a few weeks ago, is that the way cells crawl, in the case of eukaryotic cells, is by creating meshes of actin polymerization inside the cell. Those can either be in three-dimensional blebs, or what are called ruffles, in quasi two-dimensional sheets called lamellipodia, or in individual filamentous fibers called filopodia. But the key thing is that once a cell starts moving in a given direction using those meshes, it takes some time to reorient. Typically, creating a leading edge to choose a direction takes between 15 and 30 seconds. And so that means that once a cell starts crawling in a given direction, it tends to keep crawling more or less in the same direction for a period of 15, 20, 30 seconds. Some cells are more persistent and can move in the same direction for minutes. Others have slightly shorter persistence times. But it's not like a gas molecule where it's bouncing around, continually changing direction. The basic fluctuations in the Potts algorithm don't have a directionality to them. So the cell really fluctuates randomly as if it were a gas particle. And that may or may not be of significance depending on the particular simulation you're doing. And so the next exercise is really to use this concept of applied force to generate a persistent cell movement. And so what we're going to do is we're going to build eventually collective migration, because if you have multiple cells moving simultaneously, this is how you build that up. And Giuliano, do you want to take over? This is your material.
Sure. Just maybe give me 2 seconds to go there on the slides. Okay, found it. If I could grab the share. Please let me know if I'm hard to hear. I have a tendency to speak low. Yeah. Okay, so where were we? Sorry.
Just getting started. I just introduced the idea of persistent movement.
Okay, yeah, so, well, let me go forwards in the slides and then I'll go back there. Why we want persistent movement? For one, it's something that cells do a lot. They tend to move together and it's very common. Seeing that happen whenever you get a nick on your skin. Wound healing. Cells that are usually not motile, your skin cells get motile and fill in the void. And they do it in a coordinated manner. So that's the why. Now if I go back here. So now we are gonna do a bit of how we can implement collective motion in a simple way in CompuCell. And we are gonna get collective motion without the cells even directly talking to each other, even though in real life they do talk to each other. But what we are gonna do is gonna get there anyway. So we are starting a new simulation. We are gonna start with a single cell placed at the center and go from there. So make your simulation 256 by 256. You can either keep X and Y without flux, or you can use periodic boundary conditions. When I did this before, I found that having the interaction range, the pixel copy range of 3 worked a bit better. I usually actually prefer to have 2 as the range, but anyway. And yeah, that's it for here. We can use blob. And we are only gonna have, we are only gonna have one cell type. Call it cell. And then for plugins, what we are gonna want is, what we want is contact as usual. Sorry, I'm trying to lay some things here. There, now you should be seeing wizard as well. Here. So a single cell type, then no chemical fields here. And for plugins, we want contact, volume, local flex. We could use the volume flex, have it global, but it works. Center of mass, which is already selected. And we are not actually going to use cell neighbor, so no need to check that. And we are going to use the external potential local flex, because the way we are going to do coordinated motion is actually by implementing a force to the cells. And if we just change our frame of reference, we can think of the cell applying the force to itself, and it makes more sense. And that's it. Have it here. So first, we need to change the blob initializer to be a single cell. But I always find that somewhat hard to do for some reason. So instead, I'm gonna manually seed a cell at the center of the simulation by going to CC3 Python cell manipulation create cell detailed. That pastes in some code. You're seeing the presentation and to edit, right? Okay. So I'm gonna put my cell in the center of the X dimension and the center of the Y dimension. And we get those pretty easily on CompuCell. I don't need the size. Fine with, I'm gonna initialize my cell with a single pixel, and that's gonna be enough. So I have a new cell of type cell. And in here, I'm setting the cell field at the center of the simulation to be that cell. And I also need to give the cell some attributes, otherwise it's gonna pretty much disappear straight away since it's a single pixel. Like the target volume, gonna do 64 since that's what I have on the right. And lambda volume of 8. Since I'm starting with the cell being a single pixel, the cell may still disappear with a lambda volume of 8 at the start, but we'll figure that out when we get to it. And I got rid of the print cell ID loop that by default Threaded puts in the step. And one thing that you should notice is that here on the right, I have a loop over cells, and here on the left, I don't. Usually we need a loop over cells, but since I already have the cell object here, I don't need to go over the cells again. And yeah, let's just see if this shows something reasonable whenever CompCell wants to start. Okay, it's going. Okay, and my cells arrive. So happy for now with all of those parameters. Then for this single cell, we are not really gonna need NumPy, but we are gonna need it later. So I'm gonna import NumPy as NP, and then all the random number generators for NumPy are very far down. It's numpy.random, and then I'd have to give what generator I want to use. But so I'm just creating an alias for the random sub package in NumPy, calling it RNG for random number generator. This will mean less typing eventually. And now we actually have to give the cell a direction to start going in. And we are gonna first draw a random angle between 0 and 2 pi. If you want to go from -pi to pi, that's also fine. Same thing. So 2 times pi times RNG dot uniform, open close parentheses. RNG uniform by default returns a value between 0 inclusive and 1 not inclusive. So this gives you 0 to 2 pi. And to get an actual direction out of that, I'm gonna have another cell dictionary for the direction in X and the direction Y. That's gonna be the cosine and sine of that angle that we drew. We are going to need a magnitude to the force that we're going to apply. This one doesn't really need to be a cell dictionary, but since the slide has it, I'm going to do it as well. But I'm going to call it mag, less typing. And then the formula that we're going to apply to the cells, I haven't presented it yet, is this. So what we are doing here so far in those 3 lines is defining a bias direction that the cell is going to try to move towards. The cell is not going to be exact in its movement. And so the velocity and the bias are not going to be exactly aligned. They're going to be close, but not exact. So then what we are going to do is set the next bias to be somewhere in between the current bias and the direction the cell actually moved in. So if I go back here, and how much of each we choose, how much of the previous bias and the velocity we keep is defined by this alpha here. So let's start with an alpha of 0.99. Okay, and now we actually use this directions and this magnitude to get the cell going in a direction. And that, in case you don't have the cheat sheet like I do, it's cell constraint, apply force to cell. And you're gonna notice that there is a negative in the slide and also in the first term that got printed here. And that's because CompuCell is sets energies and the force is the negative gradient of the energy. That's just physics, and it is like that. Nothing we can do but remember. So in the x direction, it's going to be the magnitude time, well, the x direction. Then for y, it's going to be the y direction, same magnitude, and we don't have z, so you can just comment this out. Now, if I run this again, you see that the cell is moving a little bit downwards. I'm gonna make my cell bigger for your benefit. Not sure how this will affect things, but you should be able to see the cell better now. Yep. And it's still moving this way-ish, more down, I don't know. Okay, but we are not updating this. So in the step function, we are gonna apply this formula here to change the bias. So first we need to decide on a periodicity for this update. And I'm gonna do every 10 Monte Carlo steps. So not, if not MCS modulo 10. And as we can see here, we need the previous X position every 10. So another dictionary to add in the start. Grab X. Good plan B name. That's just going to be the cell Y center of mass and X center of mass. And now I can actually do the calculation every 10 Monte Carlo step. So the velocity in the x direction is the current cell position minus where it was before. Same idea for the y velocity. Um, and we are going to need so as you can see here in the formula, we are dividing by the velocity magnitude there, because bias is a unit vector, meaning its size is 1. And we should keep the bias with size 1. Otherwise, when we multiply by the force magnitude, we are changing the force magnitude, right? If we have a bias of 1, 0, meaning it's 100% in the x direction, that's fine. But if we have a bias that is 2, 0, it's still 100% in the x direction, but it effectively will double the force we apply. So I'm gonna calculate the modulus of the velocities, which is easy enough to do. It's gonna be the square root of the sum of the squares. Okay. Then I'm gonna grab 2x and y from up there. Make sure things are indented correctly. And it's gonna be alpha times itself plus one minus alpha times Vx divided by the modulus magnitude. Y. And if you notice, this is not going to work. And I can show you that it's not going to work. Because I forgot I had to actually put in a loop when we are in the step function. So for a cell in self, that's cell list. Now it goes. It's still mostly moving in the same direction since alpha is 99. But now we can play a bit with alpha and see what happens. So, oh, no, actually before that, something that I forgot yesterday and I'm forgetting now. Okay, I'm updating the bias. I still haven't updated the force. So I just need to copy those two lines and I don't even need to change anything. And now the force is being updated. Actually, one final thing is to update the previous X and Y. And now the cell should be updating its bias. Okay, so now let's take a few minutes for you guys to implement the plots for, you could probably do the trajectory of the cell. So plot X versus Y of the center of mass. What else? Velocity versus time. Velocity versus the target volume, lambda volume. That's gonna be for homework probably. Because maybe you saw it, but my cell, after I made it big, is moving more slowly than when it was small, at least to my eyes, which are human and are not good at estimating. Another good one to plot is the mean square displacement, but that should probably go on the homework. That way you have some time to search what the hell is mean square displacement. But with that plot, this bullet point here, do you see a switch from random to ballistic movement? It's pretty obvious. And yeah, so bullet point 1 is to change the magnitude of the force every 300 Monte Carlo steps and see what happens.
Giuliano, can you show a little bit your code on the cell dig X right there when we're changing the direction or updating the direction? Just the very end. It's cut in the, yes, right there. Thank you.
I don't know, Juliana, if you want to add the plot for the heading direction versus time.
Yep.
That might be worthwhile. And then maybe when people work on it a little bit, and then we probably should try the collective motion one too.
Yep. So, okay, I'm gonna add the plot of the heading direction and then I'll walk you through how.
I didn't use periodic boundary conditions and then it gets stuck and it stopped.
Should probably add a wall cell type and add a wall to the boundary. Make it be repulsive. Walk you through that. Cell type. Please. No. No. Wall should be frozen. Wait, what? No. No. There we go. Of course, since I have a new cell type, I have to do all the permutations of the new cell type with the cell types that are already there, or I go here, plugins, contact, replace. And then I'm gonna make the wall pretty repulsive to the cell, 25. And now to actually build a wall, Python, first thing, CCD Python, utilities, I think. Nope. Yeah, simulation, build wall. Wall. That's it. Ah, yes. I'll get to that error later.
I have a question. When you create the wall, is it considered a single cell? And when you pass through the list, does it count as? A
Yeah,
single cell?
so
Okay.
the wall is a single cell that goes around all of the domain and is only 1 pixel deep.
Mhm.
And yes, it is on the cell list, and that's why I got this error. So I'm replacing the cell list by cell list by type. That should be it. Yep. And also, since the wall is only 1 pixel, it makes sense to use even higher J's because of the contact interaction energy, right? When we have two cells side by side, they don't only see their surface, each other's surface. Right now we have a neighbor order of 4. So the cell, 2, 3, this 2 cells by each other are gonna see 2 pixels deep into each other. But the cell is only 1 pixel. So have to compensate for that. Yeah, and we see that the bias updates, well, it was going up, hit the wall, and now the bias is going to the right.
So somehow the angle, Juliana, the angle can't be right.
It's the angle divided by pi.
No, but it still can't be right because it's moving in a constant direction, but the bias angle is changing. I think you're updating one of the delta X and not delta Y or something like that.
But it's still fine for it not being exactly to the right and it moving to the right because there was the wall there.
Let's see, dir X and dir Y, how are they defined? Dir X is the force vector.
The bias. Yep.
And I would think that if you're moving along the wall, the force vector would be parallel to the wall. And therefore the direction wouldn't be changing.
The velocity is going to be parallel to the wall.
Yeah, but
And
the velocity and the force will eventually line up.
Eventually, yes. And it did. And then it hit another wall.
Okay, so that is right. So you have a long persistence time here.
Yes, it's 99.
That makes sense.
Yep.
Okay, were people able to get that to work?
Yeah, mine are still getting, I don't know, freezes and but yeah, it's working. I try to get more energy, contact energy bigger between cell and wall to avoid.
This one really works better if you use periodic boundary conditions.
Yeah, but then you don't, it's less obvious that the bias is updating because you're never heating something.
You want to introduce the collective motion? Won't have time to go through it, but you could get it started.
Yep, let's do that. No more exercises, but let's just go over the whys. So as I said, this happens in real life. I got some happening in my hand right now. And the exercise for next week or maybe homework is to do it with several cells. We are not going to do this image of the immune system arriving, doing stuff, regrowth of capillaries, scarring, etc. We're going to do this, which is a scratch assay, which is the name tells everything you need to know. You have a plate of cells. They have this little device that they scratch the center. Okay. Speed 2 times. Yeah, it's pretty literal. What they do. This. Thank you, YouTube. And this is how that looks in real life. And we can see sometimes that not only are the cells moving, but they are also growing and replicating. And actually having growth and replication is really important for this to work. Otherwise, I mean, you're going to have less density of cells otherwise, right? Something has to come here. And you may wonder how our simple model of just updating the bias is going to get us collective motion. And the reason is pretty simple. Just as the cell hit the wall here and changed its direction, when it hits another cell and it can't go there, it's going to change its direction to where it can go. And in the end, that's going to become collective motion. If you search for boids, B-O-I-D-S, bird-like objects, I think, you're going to see a more elaborate version of this equation that explicitly takes into account neighbors, and it's widely used in several instances, including the opening sequence of Tim Burton's Batman with the penguins and the bats. Boids.
I have a question. I think we talked beforehand how the cells on CompuCell are squishy.
Mhm.
Does that affect the calculation there? Because one might think that, you know, the force moving towards the other cell is not the same since it's more squishy than a normal cell.
Well, normal cells are squishy. as
In the
well.
same, oh, okay, in the
Well-
same degree or close enough.
Yeah, depends on your lambda, right? And the cell as well.
Gotcha. Okay.
But yeah, it's not gonna be like Newton's grade or anything, but
hmm The transfer of energy will still happen. Okay.
I think if you look at a classic Boyd's model, you need a bunch of rules to get things to align.
mhm.
They basically, the cells have, the Boyds have to be able to see each other at a distance. They have to maintain an equilibrium distance from each other. They have to co-align, actively co-align their orientation. But all of those things are rather artificial. Here, all you need is that the individual cells are persistent in their movement. The distance is maintained by cell adhesion and by the fact that the cells have a volume. You don't need anything more than that. The co-alignment happens automatically because two cells can't occupy the same space. So if they try to move into each other, they bend. And then the fact that they tend to move in the same direction automatically co-aligns it. And so instead of three artificial rules, you have one rule that actually we know is biologically meaningful. And so the result is, in a sense, a much more natural one. And you can get all of these kinds of behaviors that you're seeing here to happen in the CompuCell cell simulation with only that single rule. And so to me, that's what's interesting about it. The fact that you've moved from a point-like particle to an extended particle is enough to give you the behaviors you want. So I don't know, Giuliano, if you want to go through that a little bit more.
Yep.
I mean, one thing we didn't put into these slides, which perhaps we should have, is if you see the cells, the biological cells are elongated. And if you include that elongation, which you can do, not as much as you see there, but include the elongation, I think that also makes it easier for the cells to line up.
Mhm. Yep. And I don't know, spoilers that I can give you is we are gonna use the uniform initializer to have the two rectangles of cells. Easy enough to have two regions in the XML. What our force application of force is abstracting from real life and the persistence of that force is the actin polarization of the cells, and that takes a while to be rearranged. It's pretty stiff. And yeah, cell motion is complicated. Lots of papers about it. You can go in that direction if you want. But our simple version is good enough for the class. Yeah, that's it. The rest is going over how to do it.
I don't know whether you want to take 5 minutes to show them a little bit more so that they sort of have an idea of how to do it.
Sure.
And
Yeah,
then
why
we've
not?
got 5 minutes left for the class.
Okay, so changes. I'm not initializing the cell by hand anymore. That goes. Gonna remove the blob initializer since I want the uniform initializer. That's a steppable. I only want cells there. I prefer them to be 8 wide instead of 7 wide. And I want 2 regions. Both of them go from all the way top to bottom. So 0y to max y. One of them goes from 0 to halfway-ish, so 100. And the other goes from halfway-ish to the end. So let's make it asymmetrical. 150. There. Now I need to give all of my cells all of this. 64 instead of 56 there. Then instead of plotting the angle, what I probably want, you're gonna have to figure out the angle plot. What to do with that. If you do the average, which is gonna be 0 since it's a circle, or what? You probably should add a plot of all of the trajectories. That's gonna look pretty. And yeah, see, the average is gonna be 0 as promised. And we see that there is some motion and there seems to be some coordination. I don't know why they are so squarish.
Did it start the motion on the back since it has a contact energy with the wall, pushing
It
them up?
probably did. Let me just remove the plot. It's probably hogging resources. And yeah, one thing that I had seen, and I think, yeah, there is more perturbation in the back and the sides because of the wall. And we see kind of a wave going. Not so much on the left side though. I wonder why.
Is it okay if instead of 256, we do a smaller scale?
Mhm.
So we have smaller, less cells.
Yep, can do that.
Okay.
And yeah, so a few other things that cells do when they wound heal, they decrease their adhesion to each other, make it easier to fill the void and also for immune cells to arrive and move about. And as I mentioned, they grow. And we can clearly see that they are already trying to fill the void because that's pretty much the only place they can move to. But they are too held back by their neighbors and they can't fill this void. This void is going to move towards the back or the center, but there's always going to be a void. So you need cell replication there as well to get this to work.
In biology, so the cell just want to stay together, and whenever they see that there is nothing at their neighborhood, they just start moving towards the other ones? Although, as you explained, there's also inflammation, right? After a cut or something, water is put in there so they get loose. Yeah, okay.
Mhm.
But this is, I mean, I don't want to take too much time now, but this is, this simulation needs some work. This is one we need to clean up. Because in reality, what you see is that the cells in the front near the gap move inward. And if the cells in front were pulling the cells behind them passively, you'd see that the velocity of the cells at the front would be high. And then as you went further back, the velocity towards the middle would go down because you'd basically be stretching the tissue. So the cells at the edge that are moving the most would move fastest. But you don't see that. What you see actually is that the velocity of the cells doesn't decrease very much as you go away from the edge. And what that means is that the cells in the front are sending a signal to the cells behind them saying, move in the same direction. And so ultimately, the aim of this was to create that, where the movement of the cells at the front causes the cells behind them to move. Their movement causes the force on the cell. And then that force generates a force on the neighboring cells. And so in that case, you want the memory to be short because you want the cells to then actively move in the direction they were just moving. And again, this is Giuliano's simulation, so I can't speak for how he did it. But when I did this a long time ago, in the version I did, it worked pretty well. It does require some fine tuning, but you can actually get all the cells to close up pretty well. But you do have to pick the time scales and the forces pretty carefully. Okay, and anything else you wanted to show tonight, Giuliano? You could definitely get those boards like flocks.
Mm-hmm.
That's actually pretty easy to do. And that's interesting in its own right. Not as a simulation of wound healing. But, so there are a bunch of simulations and experiments one can do from this that are pretty interesting. Any other questions before we break? Okay, well, thank you, Giuliano, for taking over for that. And look forward to seeing everybody next week.